gives me extreme pleasure to follow Jude Samolsky. He's one of the pioneers of AAV research, and I hope to one day just present a similar sort of talk, describing historical perspective and mixed with science. But my dream is to one day make an impact on a field. But if I can only just achieve a fraction of that, then that would be great for me. If you told me that he had a contribution to constructing those pyramids that he showed, I would. So, what I was going to talk about today was the concept of heterogeneity, right? So, when I first started in the field, my belief was whatever ends up being packaged into AAV should be faithful to the design, right? But obviously that's not true. You can package a host of a variety of different DNA elements that can originate from the plasmids that are used during the packaging step, as well as genomic fragments coming from the host cell that's used, right? In addition, you can also get truncated genomes. And let's see if I can do this laser thing. And so a few years ago, we were very much interested in trying to understand what this heterogeneity is. And so we had developed a methodology based on PacBio's single molecule real-time sequencing in order to profile this heterogeneity. At the time, the PacBio methodology had just come online too, and it was really focused on sequencing large genomes, right? Predominantly, the Illumina platform was really the sequencing platform that everyone was using. But it fell short, right? Because in order to understand capsid composition, vector genome composition that's packaged into capsids, you would have to reconstruct this bioinformatically. But because PacBio is a long-read sequencing technology, this was able to help overcome a lot of these limitations. But one of the demands that this technology requires is that it must adapt your double-stranded material, right? So it's not an issue for single-strand AAVs, which Jude gave a very good introduction of, but in order to sequence single-strand AAVs, you would have to rely on this concept of strand annealing, right? So we know that AAV is packaged at equal ratios plus and minus stranded genomes. And so once you have these adapted by these Bell adapters, then you can have these circular, single-strand circular DNA templates, which are ideal for the PacBio platform. And so what you can do is you can, after obtaining circular consensus sequences, you can bioinformatically separate those strands so that you can understand the composition of the minus and plus strand DNAs. So with this methodology, you can obtain a lot of information, right? So number one, you can obviously understand where truncation hotspots may occur, right? So this may be dependent on the particular design, right? If you have hairpin structures that can stall replication during the packaging steps, these can form truncation events. And Wei Dong and Jude's groups had also shown that any particular DNA damage can also result in truncation events. The other thing you can do is within the same sequencing library pool, you can understand what are the contaminants that can come with your packaged vector, right? And in addition, you can also understand the distribution of this heterogeneity if you just simply graphed out the vector genome lengths, right? And you can understand how full-length genomes are packaged versus those that represent truncated or heterogeneous forms. And so this is an IGV display of an extreme case, right? So what we did was we designed a construct that carried two guide RNA cassettes that were in a tail-to-tail configuration. So this would this would form a strong hairpin sequence, which we predicted would yield a high degree of heterogeneity. So this is what you see if you run alkaline gels of this. You see multiple bands, right? But when this vector was tested in cells, it turned out that the vectors were able to work, right? So the guide RNAs were properly expressed. The two targets at these guides were designed for were able to be edited, and you also have expression of M cherry. So the question was really, how can this occur if you have such a large degree of heterogeneity, yet the gene therapy platform was working in vitro? But it turned out, based on this evaluation, that you can basically package individual components of this cassette in different particles, right? And so one of the things that makes this methodology powerful is that you can help to validate the vector designs, right? Even though you have a high degree of heterogeneity due to design. But I would argue the other gain from this is that you can take these outcomes to redesign vectors so that they are more homogeneous, right? And can confer the function that you designed them to have. So using this technology, we were able to at least validate some of the hypothesized configurations that can be packaged into AAV. Of course, you can observe full-length genomes or single-strand full-length genomes, as well as capture self-complementary forms. The other thing, as I mentioned before, is you can capture and characterize truncated snapback genomes, as well as truncated vectors that have single ITRs. Now, these are a little less represented because of the way that the methodology still relies on adapter double-stranded molecules, but these can still be identified at low percentages. The other thing you can identify are oversized dimers, right? So one of the things that was mentioned by Jude is that if you have mutated ITRs or ITRs that remain unresolved, then you can package self-complementary sequences, and these can be identified as well as oversized dimers. In relation to this, you can also detect oversized partial genome sequences, which is typically read out by these truncated unresolved ITRs. And I'll discuss a little bit about how these can be interpreted in subsequent slides. The other species that can be identified are those that have internal deletions, right? And these can occur through directed repeat sequences that can then drive large deletions from AAV genomes. And one class of AAV vector contaminants that are identified at high percentages are packaging of backbone sequences. So these may relate to the ampicillin gene and origins of replication that typically occur because of this phenomenon of reverse packaging that happens, or read-through genomes, where you have rep that reads through the ITR and into the plasmid backbone, and those components get packaged. The other thing that you can identify are other fragments, right, that can end up in the capsid that may have components that are similar to ITR sequences, which carry the packaging signal, or they may be packaged by passive packaging. One component of vector genomes that can be identified are the presence of chimeric genomes. So chimeric genomes are those genomes that carry components of ITRs that are then recombined with contaminating sequences that, again, may originate from the host cell genome or plasmid sequences used in the packaging system. And lastly, what can be detected are also adventitious viruses, specifically replication competent AAV. So because we can take advantage of the fact that you can obtain the full-length sequence of AAV that's packaged, you can then query whether a vector genome has components of both rep and cap that is then flanked or recombined with ITR sequences, indicating this potential contaminant in vector preps. And so a lot of what we now know, at least for my group, understand about AAV heterogeneity was actually learned from a collaboration that we had with Magali Budlow and Edward Aoso. So they were interested in a particular aspect of packaging AAV in SF9 insect cells. So what I'm showing you here is a native gel where we compare a 3.3 kb construct that contains a CMV promoter that's driving GFP. When packaged in HEK293 triple transfection, the triple transfection platform, it yields this very tight 3.3 kb band. However, when this vector construct is packaged with SF9 cells using the recombinant baculovirus, you can see that you have a multiple laddering pattern, suggesting that there's a high degree of heterogeneity. when you run these vector genomes in an alkaline gel, what's interesting is that you see in the insect cell-produced vectors, you observe genomes that are actually oversized, right? So what they wanted to know was the composition of these heterogeneous forms, and so we collaborated with them to run their vectors through the PacBio single molecule real-time sequencing platform, and what we were able to see confirms what was revealed by agarose and alkaline, native agarose gels as well as alkaline gels, in that the genomes packaged with HEK was fairly homogeneous, yielding 3.3 kb genomes. However, the ones that were packaged with HEK2 with the SF9 insect cells, we saw a high degree of heterogeneity. Okay, but it turns out that this phenomenon may be due to how SF9 actually packages AAVs, right? And so I'll get a little into this here. One of the ways that we were able to reveal, right, how heterogeneity accumulates in SF9 produced vectors is the way that we believe that the AAV vector genomes the libraries are designed, right? So again, one of the requirements is adaptering of double-stranded molecules, right? And so one of the steps that's required for the PacBio library build is inclusion of DNA repair enzymes. And it's speculated that these repair enzymes may be able to nick single-strand DNAs to yield double-stranded material that can then be adapted. So again, this is a truncated double-strand form that has an unresolved ITR. So these structures can actually be revealed if you observe the start and end positions, where these specific peaks can now be identified to show that to reveal genomes that are partial oversized. So one of the things that we were able to reveal through this investigation is that when packaging SF9 genomes, if it is designed as an undersized genome, that it will force packaging until it reaches the packaging step. And so one of our hypotheses is that this is the reason why SF9 produced vectors may have a high degree of heterogeneity. So one key method to overcoming this may be just to design SF9 produced vectors so that they are near 4.7 kb, the maximum packaging length size of AAV, or package half size so that you can take advantage of the fact that SF9s fail at this, right? So one of the other questions is sort of related to ITR biology and why we see these sort of events in SF9. So I don't have to go into too much detail about what ITRs are, since Jude gave a wonderful introduction. But they are composed of two configurations, right, flip and flop, that are defined by whether a domain called the B domain of the T-shaped hairpin is closer to the open end, which is referred to flip, or a configuration where the C arm is closer to the open end, and this is referred to as flop. AAV is unique from other parvoviruses because they package flip and flop at each of the ends at 1-to-1 ratios, and this is because of the way that AAV replicates, right? So after it's rescued from the plasmid, and this mechanism for rescue is not really that well understood, but once you have a rescued single-strand species, this then becomes the replicative form, right? So we all know that DNA replicates 3' to 5', so when the second strand is then synthesized, flop-oriented molecules will always replicate flop. But what changes the orientation is the fact that upon nicking by rep, the old 3' ITR becomes the new 5' end. And so this changes the orientation. So the outcome of the first replication cycle would then be a flip-flop molecule that is plus-stranded, and then the flop flip molecule that is minus stranded. And so if you do this a few times, then you can actually encompass all 8 configurations, representing all of the 1-to-1-to-1 ratio, 1-to-1-to-1-to-1 ratio of flip, flip, flop, flop, flip, flop, and flop, flip. And so if you ever sequence AAVs and you can define this composition, then you can actually obtain some information about how well the AAV genome is replicated. Because when we compare AAV genomes that are packaged by HEK293 cells, what we see is that at both the left and right ITRs, they have nearly equal ratios of flip and flop. However, when we look at the composition of the ITRs of the SF9 produced vectors, we see that majority of them are unresolved, but in addition, we see a lot of mutated ITRs that contain missing B arms or missing C arms. We even see genomes that have trident-shaped ITRs. And we were curious about what these ITRs were doing, right? Perhaps the high degree of mutated ITRs were leading to these unresolved genomes, right? Giving you this sort of a high degree of heterogeneity in packaged genomes. And so what we were able to do was to stratify the ITRs in both HEK293s and the SF9 produced vectors in unresolved, for those that carry unresolved ITRs, those that were trident shaped, as well as missing B or C arm. And what we were able to see was that all of these ITRs that were mutated within both systems had a high degree of these unresolved ITRs. So the suggestion is that the reason why SF9 vectors produce a high degree of heterogeneity is because of the simple fact that they have a higher degree of mutated ITRs. While for HEKs, they do also have mutated ITRs that do have issues with ITR resolution, but predominantly so, most of them are wild type, and they can package the full flip and flop composed genomes. And so one of the questions that I always get is that when they look at their sequences, they can see genomes with multiple ITRs. So if you had a genome that contained mutated ITRs that failed to resolve, right, you can imagine that for every cycle that it undergoes for replicating the gene of interest, that if it fails to resolve the ITR, then you can get just continuous rolling hairpin replication, right? And at the end, you can get these sort of gigantic molecules. Now, if you imagine that you have a genome that is truncated and just contains a partial sequence that fails to undergo proper resolution, you can have these molecules that contain multiple ITRs. And so these may be these are highly likely the reason. This is highly likely the reason why you see these sort of compositions in your reads. And so I would argue that these aren't so much a sequencing artifact, but it may represent what's truly being packaged into AAV. And so one of the things that I wanted to bring attention to is the observation of chimeric genomes. And so as I mentioned before, chimeric genomes are acquired by recombination that occurs during the packaging step, where you carry different components. And so, for example, the AAV vector genome that I'm showing here on the bottom contains 6 different regions, stretching from the human chromosome 5 sequence to ampicillin resistance gene sequence, as well as sequences that encapsulate capsid DNA, right? And so one of the points that I wanted to sort of drive home with highlighting these chimeric genomes is that for the majority of those sequences that we see are co-mapping to multiple references, which is how we identify these chimeric genomes, they typically also co-map to the vector genome sequence. And they for the majority of the cases, they also retain or can map to ITR sequences that are found in the vector GOI reference. And so what this suggests is that a lot of the contaminants that we see also contain ITR elements, which provide the driving mechanism for packaging, right? So this sort of data would suggest that packaging of these contaminants isn't passive, but is actually based on the presence of the ITRs that are found within them. But I would note that a lot of people that have come to me to describe their chimeric genomes, that they don't see ITRs. But what we've been able to show, right, and I'm not showing here in these slides, but a lot of these co-mapping studies, they're really dependent on the mapping tools that you use. So what we've been able to show was that certain aligners don't do as great a job as identifying short reads, right, that may confer the presence of chimerics. And the other thing that's very important is to really have an understanding of what the composition of the GOI, because a lot of the vectors that may be shared may still have remnants of sequences that may match the human genome, right? So when you're doing these sort of investigations and you find that there is a high degree of mapping to a certain portion of the human genome, it could very well be that they may be part of the vector genome. And those sequences need to be masked, right? So when identifying and characterizing chimeric genomes, a lot of care and effort has to be made in order to do these investigations correctly. So I wanted to end with this slide because I like this idea that chimeras are defined as fire-breathing monsters from Greek mythology, but the other sort of definition that I also enjoy is that it is also an illusion or fabrication of the mind, right? But one of the things that I wanted to highlight is the fact that these chimerics that we're identifying, they're real, right? So they're giving us an indication for how heterogeneity can be defined and ought to be characterized in our preps. Because if these are driving forces for certain levels of toxicity that we see in patients that are being treated with AAV, then they certainly need to be understood so that we can decrease the presence of these recombination events as well as what is being packaged, right? And with that, I'll conclude with my acknowledgement slide. I'd like to thank members of my group, Tam Tran, Mitch, and Suk, who are the 3 individuals who have been focused on this work, as well as our sequencing core at UMass Chan Medical School, and PAC Biosciences, who've been very key to leading this research to develop these tools for us. And so, and Magley's group at University of Donat. And I can take questions. Thank you.